Hello students, in this video we shall see the solutions for the NEET test 4 which was conducted on 16th of September. First question, the bond formed between carboxyl group of one amino acid and amino group of the adjacent amino acid. So the amino acid structure if we see, so it will have this structure. So this is the carboxylic acid group COOH and the adjacent one NH2. NH2 is amine group. So here this is the amine group. So these two groups, they will combine to form peptide bond. They will form CONH bond by the loss of water. Find out the wrongly matched pair. So if we see here, primary metabolite ribose, that is correct. Secondary metabolite is anthocyanin, that is also correct. Protein is insulin. Insulin is the protein hormone. Cellulose heteropolymer. It is not the heteropolymer. It is the homopolymer. So it is homopolymer of glucose. So the answer is option D. Third one, pentoses and hexoses are common. So pentoses means which will have 5 carbon in one sugar unit, and hexoses means they will have 6 carbon in one sugar unit. So they will have only one sugar unit. So they are monosaccharides. Then fourth one, if all the peptide bonds of a proteins are broken, then the remaining part is. So we know the protein is polymer and the monomer is amino acids. So it's like this. So each monomer. So this is the amino acid. So they are linked by the bond. So these bonds are peptide bonds. When peptide bonds are broken, we will get amino acid. Then fifth one, arrange the steps of catalytic action of an enzyme in order and choose the correct option. So here the enzyme action, they have given the steps. So first what happens, enzyme and substrate will bind, and then they will form enzyme substrate complex, and then they will form enzyme product complex, and then the enzyme will get separated and product will get separated. So this is the reaction which will be happening. This is the action. So if you see here, out of the given one, first one is the fourth one. Substrate binds to active site of the enzyme. So this is the first step. Then binding of substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. This is the second step. The active site of enzyme is in close proximity of the substrate and breaks chemical bonds of the substrate. This is the third one. Then enzyme releases the product of the reaction and the enzyme is free for another substrate. This is the last step. So the answer is option A. 4, 3, 2, 1. Then sixth one, maltose gave rise to 2 molecules of. So again, maltose is a disaccharide. It contains 2 sugar units and the 2 sugar units are glucose. So 2 molecules of glucose, they will form maltose. Then seventh one, feedback inhibition of enzyme is affected by which of the following? So we know feedback inhibition in the sense in a reaction, step by step reaction, the end product or the intermediates, they will inhibit the action. So the reaction will be inhibited. So answer will be option D. Eighth one, globular proteins undergo structural changes in response to extreme pH or temperature. When there is extreme pH or temperature, the structure changes, and that process we call it as denaturation. The ninth one, starch can be used as an indicator for the detection of traces of. So again, when there is starch, starch has a helical structure. So in this helix, it will hold iodine molecules. So because it can hold iodine molecules, it will give blue-black color. That means it can detect if iodine is present, then it gives blue-black color. If there is no iodine, it will not give any color. So this can detect the presence of iodine. The 10th one, which one of the following structural formulae of two organic compounds is correctly identified along with its related function? So the first one, A, is the structure of lecithin, and B is the structure of adenine. So now we shall see. So A cannot be the answer because the structure is only not identified properly. Even B cannot be the answer. A, C, and D option identification is correct. So the explanation, lecithin, a component of cell membrane, it is correct because lecithin, it is a phospholipid, and this phospholipid, it forms the cell membrane. Adenine, a nucleotide that makes up nucleic acid. It is not nucleotide, it is just the nitrogen base. So the answer is option C. 11th one. Which of the following is a disaccharide? So out of here, glucose, fructose, galactose. So these are monosaccharides. Sucrose is disaccharide. So sucrose is made up of glucose and fructose. Then 12th one. Example of a typical homopolysaccharide. So lignin and suberin, they are the heteropolysaccharide. Insulin, it is a protein. Starch, yes, it is a homopolysaccharide of glucose. Then 13th one, enzyme often have additional parts in their structures that are made of molecules other than proteins. So they are called cofactors. Where this additional chemical part is an organic molecule. So if it is organic molecule, it can be either prosthetic group or it can be coenzyme. And the other type of cofactor is metal ion. Metal ion is inorganic. So answer will be option B, coenzyme. 14th one. Given below are the two statements A and B. Choose the correct answers related to the statements. So here, first statement, amino acids are amphoteric in their function. Yes. So amphoteric means they can act like both acid and base. Why they can act as both acid and base? Because they have these two functional group, carboxylic acid and NH2 group, and both can be ionizable. They can form ionized structure, right? Because of this, they are called as amphoteric. That is a correct statement. Then statement B, all amino acids are necessary for our body. No, we have different. We have two types. One is essential amino acids, and we have non-essential amino acids. Right? Essential means it is synthesized in our body, so no need to take from external source. The non-essential means it is synthesized in our body and no need to take from external source. So the second statement is wrong. So A is correct, but B is wrong. Option A is the answer. Then 15, holoenzyme. So we know holoenzyme is the functional enzyme, the activated enzyme. This holoenzyme contains protein part, and this protein part we call it as apo enzyme, and we have non-protein part. And this non-protein part is called as cofactor. So your answer is non-protein and apo enzyme. Then 16, select the wrong statement. So we shall see one by one. Proteins are heteropolymers made of amino acids, which is correct. There are 20 different amino acids which will be present in a protein. Then ribozymes are nucleic acid with catalytic power. Yes, so they are the nucleic acid. Exactly, they are RNA which has the catalytic power. Nucleic acids serve as genetic materials. Both RNA and DNA, they serve as genetic material. Collagen is the most abundant protein in the whole biosphere, which is wrong. Rubisco is the abundant protein in biosphere, and Rubisco is the most abundant protein in animal world, which is wrong. In animal world, it is collagen. So they have given ulta. So this is the wrong statement and the correct answer. 17. Lactose is composed of following components. Again, lactose is disaccharides. It is made up of glucose and galactose. 18. The lock and key model of enzyme action illustrates that a particular enzyme molecule. So again, lock and key model if we see. So if this is an substrate, so the enzyme, substrate and enzyme will have exactly the complementary shape. So that means they are specific. They are the interaction is specific. The specificity between enzyme and substrate is being explained by lock and key model. So here option B says that interacts with a specific type of substrate molecule. That is the answer. Then 19. So here the curve given below shows enzymatic activity with relation to three conditions: pH, temperature, and substrate concentration. What do the two axes X and Y represent? So again, always x-axis there it will be either temperature or pH or substrate concentration, and y-axis will always be enzyme activity. So here if we see option A, so the option A is correct. And even B, you think B can also be the answer, but for if substrate concentration is in x-axis, the graph will be like this. So the graph is like this, so it should be temperature or pH. Then 20. Enzymes having different molecular arrangement but similar function. So such kind of enzyme we call it as isoenzyme. 21. Choose the type of enzyme involved in the following reaction. So here you can see there is transfer of this functional group. So this group is with S. So if you see the product side, it is with S dash. So this is done by the enzyme transferase. Then the bond present between carbohydrate molecule, it is a direct question, glycosidic bond. 23. One strand of DNA has the sequence of nucleotide, this, then other strand of DNA has. So again, we have to see the complementary basis. Both the strands are complementary. So for 3 prime, it will be 5 prime. For in the place of for A, there will be T pairing for TA, and for CG will be pairing for GC. So this will be the sequence, and this is in option D. Pick out the wrong statement. So first one, amino acids are substituted methane, which is correct statement. Glycerol is a trihydroxypropane, which is also correct. Glycerol contains 3 OH groups and 3 carbon atoms. So, glycerol is trihydroxypropane. Lysine is a neutral amino acid, which is wrong. Lysine is acidic amino acid. Lecithin is a phospholipid. Yes, it is a lipid and it contains phosphate group. So, it is a phospholipid. So, the answer is option C. Then 25. One feature common to all the compounds found in the acid soluble pool is. So in the acid soluble pool, there will be micromolecules. And these micromolecules molecular weight will be less than 1000 Dalton. So that means it will be around 18 to 800 Dalton. So you can see here option A and C, they gave these term 18 to 800, but Dalton is mentioned only in option A. So here in C, they have not mentioned anything. Dalton, nothing is mentioned. That means when there is nothing mentioned, we can't say what it is. It can be kgs, it can be grams, anything it can be. So unit is must and should. We have to note it. So answer will be option A. Given below is a structural formula. So this is a structural formula of ribose, and ribose is pentose sugar. 5 carbon atoms. So 1, 2, 3, here 1, 4, and this is 1, 5 carbon atoms. 27. Where the starch is stored in plant cell. So we know in the plastids, so there are 3 types: chloroplast, chromoplast, and leucoplast. So again in leucoplast, we have a type of leucoplast based on the storage. So alluroplast, amyloplast, elioplast, we have different types. Among them, amyloplast will store starch. Then 28. Amino acids are the substituted methanes. Name the 4 substituent groups occupying the 4 valency. So the 4 valency are, so one is the variable group, hydrogen group, carboxylic acid group, and the other is amino group. So this is in option A. 29. The amino acids are acidic, basic, and neutral based on the number of. So again, here the structure of amino acids. So if we see this amino acid structure, so this amino group is basic and this carboxylic acid group is acidic. So right now it is neutral. So it depends on the R group. So R group, if it contains NH2 group, the entire amino acid is basic. So here, if it contains carboxylic acid group, it will become acidic. So on what basis we are saying acidic basic? On the basis of amino group and carboxyl groups. Then 30. Identify the term ash in the term of living tissue sample analysis from the statements given below. So here ash in the sense, so once we get the dry tissue and when we burn it, all the carbon containing compound will evaporate and the other molecules, the inorganic molecules will just burn, and that is the ash. It contains inorganic elements like calcium, magnesium. So this is the B is the answer. Then 31. Amino acids are organic compounds and are called alpha amino acids. Why? So why amino acids are called as alpha amino acids? Because both the groups. So here in this structure, both amine group and carboxylic group, they are attached to the same carbon atom. So they are called as alpha amino acids. So this is given in option B. Then 32. Variety of amino acids are formed based on nature of R group. R is the variable group. So variable means only this group will be varying. So on the basis of that, we have different types of amino acids. Identify in which of the following compound heterocyclic rings can be found. So heterocyclic rings we can find only in nitrogen bases. Proteins, amino acids, lipids, we do not find any rings. Phospholipids are, so here we have phosphate group, which is the inorganic part, and we have lipids. Lipids is the organic. So here lipids is associated with phosphate. Such lipids we call it as conjugated lipids. Then, so some structures of amino acids are given. We have to identify it properly. So A is alanine, B is glycine, and C is serine. So answer is B. 36. Identify the Zwitter ionic form in the given reversible reaction. So Zwitter ionic form in the sense both amine group and carboxylic acid group should be present in the ionic form. So here only amine group is there. In C only carboxylic acid group is ionic. In B both are ionic. So B is Zwitter ionic form. 37. The physical and the chemical composition of amino acids are essentially of the again physical and chemical composition. It will depend on all the functional groups. So here we have amino group, carboxylic acid group, and R group. So based on all these three functional group, the physical and chemical composition of amino acid will be dependent. Then 38. Biomolecules are constantly being changed into some other biomolecules and are made from. So it is the turnover. So one biomolecule will be converted to other, and that biomolecule will be converted to some other biomolecules. It's like one biomolecule give rise to other biomolecule. So it is option B. The most important form of energy currency. So energy currency, it is always ATP, adenosine triphosphate, which is a nucleotide. Then 40. Proteins with catalytic power. So these are catalytic power in the sense which will enhance the rate of the reaction, that we call it as enzymes. 41. Why metabolic pathways are comparable to automobile traffic in a city? So again, if we take any metabolic pathway, they have a definite direction. Only in that particular direction they will be going on. And if we consider automobile traffic, so all the vehicles, they'll be moving in a particular direction. They do not travel haphazardly. So there is a particular lane in which they will be moving. So that is why, since they both have a particular direction, metabolic pathways are comparable to automobile traffic. Next question number 42. Pick the odd statement. So that means again, which statement is not correct. So we'll go one by one. Removal of carbon dioxide from amino acid converts amino acid into an amine, which is correct. Only amino group will be present. Only NH2 functional group will remain. So it is an amine. All the biomolecules have a turnover, which is correct. We have just seen it before 2-3 questions. Metabolic pathway are termed as transformation reactions. Yes, one molecule will be transformed to other biomolecules, so they are transformation reactions. Metabolic pathways always follow linear route. They will always follow crisscross. They will cross with other metabolic pathways. So answer is D. 43. Those nucleic acids which behave like enzymes, they are called as ribozymes. Then 44. Enzyme catalysts differ from inorganic catalysts in which way? We'll see one by one. Enzyme catalysts are similar in size and lesser in weight in comparison to inorganic catalysts, which is wrong. Inorganic catalysts are smaller in size and lesser in weight. So they have given ulta. Inorganic catalysts can work efficiently at high temperature, yes, but enzyme cannot. Inorganic catalysts can work at high temperature, high pressure, which is right, but enzymes cannot. So that is also correct. So answer is both B and C. 45. Almost all enzymes are proteins except ribozyme. 46. The rate of the reaction doubles or increases by half for every 10 degrees Celsius. So law of Newton. Then 47. A competitive inhibitor competes with a substrate for the substrate binding site of enzyme due to it. So again, why the competitive inhibitor will compete with substrate? Because they have structural similarity. So we know this malonate and succinate. So these two are the uh, analogs. Analogs means they have similar structure. So because of their similar structure, they will compete with the active site of the enzyme. Then 48. Select the correct graph which shows the effect of temperature on the velocity of a typical enzymatic reaction. So previously also we had seen such question. So it should be option C. Then 49. Which enzyme catalyzes the breakdown of hydrogen peroxide to water and oxygen? So this will be done by peroxidase and catalase. Next 50. So here some statements are given and you have to select which is correct with regard to enzymes. So first statement, enzymes do not alter the overall change in free energy of a reaction. Yes, it is correct. Enzymes are highly specific for reaction. Yes, each enzyme is specific for substrate specific. That means they are specific for specific reactions. The energy input needed to start a chemical reaction is activation energy. This is the extra energy which the reaction requires. So to convert into product, that is activation energy. Enzymes are proteins, yes, whose three-dimensional shape, that is the 3D structure, the tertiary structure of the proteins that is necessary for their function, which is also correct. So all the statements are correct. 51. Enzymes catalyzes the biochemical reactions by, so always by lowering the activation energy, the enzyme will increase the rate of the reaction. So what are proenzymes? Proenzymes, they are the inactive form. When the proteins, enzymes are formed, they will be in inactive state. Such inactive state proteins, we call it as proenzymes. Then 53, Michaelis-Menten constant, it is equal to, it is substrate concentration. So at which rate attains half of its maximum velocity. So if this is a graph and this is substrate concentration and your enzymatic activity is given, so this is how the substrate concentration will alter. So this one is a Vmax and this is the half of Vmax. And if we extrapolate, so this substrate concentration, this is the Km value. Michaelis-Menten constant. Then 54, which is a grape sugar. Grape sugar is also called as glucose. Then 55, identify X and Y. So here the X is the first amino acid. So first amino acid at the extreme left hand, there will be N terminal, and the last amino acid is Y. So the Y end, right end will be C terminal. So answer is A. 56. Answer briefly. Which color glycogen gives on its reaction? So it will be red color. And what is satellite? So it is the repetitive base pairs. Name three components of nucleotide molecule. It is phosphoric acid, pentose sugar, and nitrogenous organic base. So it should be option B. Then 57. According to Watson-Crick model, DNA exists as, so DNA, it exists as double helix. The two strands of polynucleotide strands are, both the strands are anti-parallel. The backbone is made up of sugar. So C should be sugar, they have already mentioned. Sugar, and D should be phosphate chain. The nitrogenous bases are more or less dash to the backbone. They are perpendicular to the backbone. So if this is a backbone, they will be, bases will be perpendicular like this. So the answer is C. 58. In DNA, A base pairs with, so it pairs with T, and between A and T there will be two hydrogen bonds. One full turn of helix, it involves 10 steps. 10 base pairs will be there. Next 60. A in the given structure represents. So which bond is that? It is phosphoester bond. It is option A. 61. Identify the structural formulae and select the correct option. So your A is adenine. So your B, for this adenine, sugar molecule is added. So it should be nucleoside. Nucleoside of adenine is adenosine. So here there is adenine, sugar, and also there is phosphate group. So it is a nucleotide. So it is adenylic acid. So answer is option A. For nucleic acids, the building block is, that means the monomers are nucleotide. 63. Which of the following is incorrect? So we'll see one by one. In cellular organisms, DNA is a genetic material, yes. In all the cellular, only in acellular, that is in viruses, RNA can be genetic material also. Adenylic acid is nucleoside. No, it is a nucleotide. Just before we had seen. Cytidine is nucleoside, which is correct. Nitrogen bases, A, G, C, T, U have heterocyclic rings. All nitrogen bases has heterocyclic rings. So B is the incorrect one. Okay, so next question, 64. Which of the following statements are correct for polysaccharides? So we'll see one by one. The polysaccharide are found as a part of the acid insoluble pellet. So it should be acid insoluble part. Yes, so this is correct. These are long chains of sugar. Yes, so they are the polymers of sugars. They are threads containing different monosaccharides as building blocks. Yes, so they are the monomers of monosaccharides are the monomers of polysaccharides. Cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, fructose. It is not fructose, it should be glucose. So, except fourth one, all are correct. Adult human hemoglobin has, so human hemoglobin has 4 subunits. Out of that, 2 are alpha and 2 are beta. 66. How many carbon atoms generally take part in the formation of monosaccharides? So for monosaccharide, minimum there should be 3. It goes up to 7. 67. Select the correct which represent the homopolysaccharides made up of glucose monomers. So out of this, D options, starch, glycogen, cellulose, all are the homopolymers of glucose. Then 68, primary metabolites play an important role in. So they have the direct role in the physiological process. Then 69, the proteinaceous molecule that joins a non-proteinaceous prosthetic group to form a functional enzyme is called. Again, we had the same type of question before. It is called as holoenzyme. The proteinaceous molecule is called as apoenzyme. So holoenzyme is a functional enzyme. Then 70. A high fever is dangerous to humans because high fever in the sense the body temperature increases. It is danger because when the temperature are increased, then the enzymes will be denatured because at high temperature enzymes, the proteins will get denatured. They will lose its structure. 71. Which of the following statements are incorrect? Left end of polysaccharide is reducing, non-reducing, while right end is reducing end, which is correct. Starch and glycogen are branched molecules, also correct. Starch and glycogen are the reserve food materials in plants and animals. Starch is the reserve food in plants and glycogen is in animals, which is correct. Starch can hold iodine molecules in its helically secondary structure, but cellulose being non-helical cannot hold iodine, which is also correct. So none of the statements are wrong. All statements are correct. Proteins are heteropolymers which are made up of dash type of monomers. So if you take amino acid, totally 20 types of amino acids are present. Then 73. Which of the two groups of following formula is involved in peptide bond formation between different amino acids? So it is always this amine group and carboxylic acid group, that is 1 and 3 always. Then 74. So some pictures are given. We have to choose which is the correct one. So here A is the primary structure. B. So here 2 units are there. 2 subunits. So it should be quaternary structure. And C, it is forming hollow woolen ball like structure. So it is tertiary structure. And this is a helix, alpha helix. So secondary structure, which is given in option C. 75. And alpha helix is an example for, so just now we had seen alpha helix is the secondary structure. 76. Acidic amino acid carry 2 COOH and 1 NH2 group per molecule. Keeping this in mind, select the correct pair of acidic amino acid. So here the name itself says aspartic acid, glutamic acid, both are acidic. So answer is B. So here assertion and reasoning type. So 77. Coenzyme or metal ion that is very tightly bound to enzyme protein is called prosthetic group. This is correct. Statement 2. Complete catalytically active enzyme together with its bond prosthetic group is called apoenzyme. It's not hapoenzyme, it is holoenzyme. So it is wrong. So for 77, it should be option C. 78. Arachidonic acid is an unsaturated fatty acid, which is correct. There are one or more double bonds between carbon atoms in unsaturated fatty acids. Yes, so this is also correct. So again, why we call fatty unsaturated fatty acid? Because they have one or more double bonds. So it should be option A. In the solutions of different pH, structure of amino acid changes, which is correct because COOH and NH2 group are ionizable. So even the statement 2, what they given, that is actually the reason for statement 1. So it should be answer A. Then match the following type. Question number 80. Acid soluble pool goes with filtrate. Filtrate is acid soluble pool. Acid insoluble pool is retentate. And water goes with inorganic compound. Protein is organic compound. It should be option A. Then question number 81. So here dehydrogenases, they are also called as oxidoreductases. So this is with third one because oxidation reduction reaction happens. Then ligases. So what ligases will do? So they will link the two bonds together. So fourth one, isomerases. So they will convert one isomer to another isomer. So option 1. So it goes with first one. Hydrolases. So here hydrolysis. Spelling is mistake. Hydrolysis. So fifth one. Transferases. Transfer of groups. So it is second. So here this should have been second. So answer is D. Then 82. Prosthetic group. Example they have given. Prosthetic group is heme group. Cofactor is NAD. Sorry, cofactor. See, cofactor generally they have given. Cofactor can be any one or two or three. Coenzyme is NAD. So, but if you see the option, only the left out is third one, right? So we can go with option A. 83. So here again in the column 1, some structures are given. In column B, their components. So cellulose fiber, it is made up of cotton fiber. It is made up of cellulose. Then exoskeleton of cockroach, it is made of chitin. Then liver, it stores glycogen. Peeled potato, it will have starch. Root of dahlia will have inulin. It's not insulin. It should be inulin. So answer is option B. Then 84. Amino acid, monomer of protein. Fatty acid and glycerol, they are the components of lipids. Nucleotides, monomer of nucleic acid. Simple sugars are the monomer of polysaccharides. Answer is option A. Then 85. Again, prosthetic group, it is of the cofactors. Prosthetic group, they will tightly bound to apoenzyme. Coenzyme, they are transiently binding to apoenzyme. Metal ion, they will form coordination bonds with apoenzyme. Then assertion reasoning questions. 86. Assertion: most biomolecules which are found in acid insoluble fraction are macromolecules, which is correct. Most biomolecules found in acid insoluble fraction have more molecular weight, more than 1000. Since they have more molecular weight, they are found in acid insoluble fraction. So answer should be A. Lipids are not strictly macromolecules, they are micromolecules because their weight does not exceed 800 Dalton. And reason is the correct reason. Then 88. Last amino acid of any protein is known as C-terminal. Yes, so the extreme right end is C-terminal because there will be free carbon carboxylic acid group. So reason is also correct and it is the correct reason. Sucrose is known as invert sugar. Yes, sucrose is a disaccharide. Both the statements are correct, but not the reason. So it should be option B. Then starch can be stained with iodine, but cellulose cannot be stained by iodine, although both are homopolymer of glucose. Yes, that is correct. Cellulose do not contain complex helix, but so cannot hold iodine molecules. Starch has the helix, so it can hold iodine, but cellulose do not have helical structure, so it cannot hold iodine molecules. So even the assertion, reason, statements are correct, and reason is a correct reason. So his answer should be A. So all the questions are discussed. Please go through the video if you have any doubts. Thank you.